Diabetes is a serious condition that affects 29 million people in the United States. This project is done by Noam, Jack, and Shrey. An important part of any biological system is the homeostasis, the ability to maintain equilibrium. This keeps biological systems running smoothly. Diabetes is the disruption of the body's ability to maintain the blood sugar levels in its system, and the homeostasis is disturbed. Diabetes has to do with the disruption of insulin, the signaling protein that is important for maintaining blood sugar levels. Cleaved from a larger proinsulin, insulin is too small to be folded initially. And as you can see here, insulin is a compact protein with 3 disulfide bridges to hold it together. Now, as you can see, it looks pretty large, but in reality, it's a lot smaller than most other proteins. Now, if blood sugar levels are too high, insulin is released, and this returns the blood sugar levels back to a status of equilibrium. Insulin, the triangle, binds to the tyrosine kinase receptor protein that phosphorylates other proteins, leading to a signal transduction cascade that facilitates glucose transport out of the bloodstream and into the cell. Now, if blood sugar levels are too low, glucagon is released, converting stored glycogen in the liver into glucose, and the blood sugar level rises, maintaining equilibrium. When blood sugar levels are too high, insulin is released and blood sugar levels will decrease. But when blood sugar levels are too low, glucagon is released and blood sugar levels will rise again. This is honestly the whole mechanism. Now, type 1 diabetes is a failure of insulin production in the body, and insulin is produced in cells called the islets of Langerhans, and there are about 1 million of these in a healthy pancreas. And these islets make up 1 to 2% of any pancreas in general. Each islet is a cluster of 3,000 to 4,000 beta cells which produce insulin. In diabetes, an autoimmune response targets and kills these islets and results in no insulin production. Some symptoms can include thirst, hunger, and headaches. And these can be signs of diabetes. Hello, Gnome's hand. Blood sugar levels can be tested or tasted in blood or urine samples. Recombinant human DNA is the process of the INS gene, which is responsible for insulin production in humans, is put into bacterial plasmid so the bacteria can actually create the human insulin using their own genetic resources. The 2 main problems with this treatment is the fact that the insulin degrades over time and that the injections necessary are frequent and expensive. The future research hopes to achieve more effective and cheaper treatment methods. Thank you for watching, and goodbye.